Our third and final talk of the session will be Ricardo Rios-Carrillo. Ricardo is a PhD student also from the Brain Connectivity Lab at the Instituto de Neurobiología de la UNAM, and he'll talk about the differentiation of white matter histopathology using b-tensor encoding and machine learning. Please take it away, Ricardo.
Hi, thank you. Give me a second. Okay, can you see my slides?
Looks perfect.
So thank you for the presentation. In this work, here with a data-driven approach that uses a machine learning algorithm to solve a classification problem that aims to differentiate histopathological tissue on a model of white matter degeneration. We solve this using diffusion metrics, extract from a b-tensor encoding acquisition, and then we ask to the algorithm which metrics were the most significant features to solve this problem. We use an animal plot model of white matter histopathology in the optic nerve. With this setting is we can affect only one nerve and leave the other one intact for direct comparisons. We plan different experiments for white matter. These are just idealized illustrations of what was expected from this experiment. They represent a coronal perpendicular slice of a nerve with simple representation for axons of different sizes with myelin or glia. We hope for four patterns of white matter, control group, a group with axonal loss produced by retinal ischemia, a group with axonal loss and layer activation produced by the injection of LPS, and a group intended to measure only the mechanical damage produced by the injection of the saline solution. We also obtained toluidine blue histology to corroborate and guide our experiments. This is also a perpendicular histological slice of a healthy nerve. In this means you can observe myelinated axons of different sizes with corresponding myelin and the glia in this part. As we start analyzing our histological picture, four resulting parents of white matter emerge. An intact one, populated with myelin axons, as you can see in these circles, and glia in a normal state in these blue squares. An injured parent that presented axonal loss areas in these green circles with axonal collapse, but with some small areas with axons, these little blue circles here. And the glia also present signs of to be on a reactive amoeboid phase in these green squares. And finally, a parent that we call injured plus with total axonal loss, amoeboid glia, and even glia in a foamy state in these red squares. This is actually microglia that it's on a phagocytosis phase. These three parents were homogeneous along the whole nerve. The last four parent was heterogeneous one that have the limited regions of the other three paradigms. So this one, for example, contain a region in the intact class and a region in the injured class. So it's actually possible to manually delimitate these regions in histology. So we reframe the outcomes of our initial experiments to these new four experimental parents. Generally speaking, the controls became the intact ones. The ischemia produced consistently the injured parent. Both injections experiments produce the majority of the general and injured plus parents, depending on the mechanical damage caused by solid injection. So this serves as a friendly reminder that experiments are not always perfect, but for the purpose of this work, it's actually great because the idea here is to use the quantitative diffusion properties of the homogeneous classes to train a machine learning algorithm that properly classify each voxel region on the original parent and identify the most important diffusion features needed to solve this general problem. For the quantitative MRI measures, we obtain diffusion metrics through the b-tensor encoding acquisitions. B-tensor encoding is the dimensional extension to the typical B-vector used in the standard diffusion MRI acquisitions. The b-tensor encodes the acquisition parameters, but besides the b-val and b-vec used in the standard diffusion MRI, it also has a B-shape, which makes the diffusion MRI signal specific to certain geometries in the medium. B-tensor encoding uses multiple tensors of different shapes, sizes, and orientations to quantify the diffusion correlation that occurs in multiple directions at the same time, giving us access to new complementary information in the diffusion MRI signal that we can use for complex signal representations or microstructural modeling. The prices that the b-tensor encoding impulse program sequence aren't as simple as the square-shaped gradient waveforms used in the standard diffusion MRI acquisitions. They are not constructed by complex gradient waveforms needed to obtain the desired b-tensor. But as I said, they contain new information that can, for example, give access to new diffusion MRI metrics that describes a diffusion tensor distribution process instead of a single diffusion tensor as usually done in diffusion tensor imaging, DI. So in this work, we obtained a b-tensor encoding image in a preclinical 7 Tesla Bruker scanner for an ex vivo setting, using the sequence in the preclinical neuro-MRI developed by Microstructure. The gradient waveforms were optimized using the Now toolbox, and the acquisition protocol is modified from work from the Lung group with proper modification from our ex vivo setting. We pre-processed this image, and then we did the Q-space trajectory method in its QTI plus implementation. This is a signal representation from b-tensor encoding acquisitions that extract 8 diffusion metrics. 4 of them are akin to the ones obtained by diffusion tensor imaging, and the other 4 are intended as descriptors of a diffusion tensor distribution representation. Micro-FA is homologous to FA, but is not affected by the dispersion in the medium. CC measures the orientation coherence in the examples of the microtensors. Ki and Ka are both kurtosis sources related to the variance in sizes and shapes and anisotropy of the microtensors in the DTD. Here I show an example of the obtained contrast for a subject in this setting, and a schematic one in their injured class. Here we can see some features like FA and micro-FA. There is a clear asymmetry between the left, the control, and the right experimental nerves. We train a machine learning classification algorithm to predict the data for each voxel in the regional pairing data. It is the raw data from our experiments. Each dot here represents a voxel in the data of the optic nerve in 2 dimensions, just for the visualization. This is our trained test data. This is a representation from the regional data, the problem that we actually want to solve. Specifically, we train a random forest algorithm using the AQTI extract features and predict the regional data into probabilities to belong to each one of the homogeneous classes. We combine those probabilities back in RGB map and then project it back to the MRI space image. The next figure shows a schematic with results for 3 slices at different levels of the same subject. This is the obtained RGB mat for both nerves, the left and the right experimental one. And this is our example histologies from both nerves and approximately the same position. Comparing both, we can observe there is an excellent spatial correspondence between these topology areas here in green, in the histology and the MRI, giving evidence that the algorithm works pretty well. On the machine learning side of things, the classifier obtained an accuracy of 80% in the training set to distinguish these 3 classes. The confusion matrix shows that it's easy to distinguish between the injured class and the injured ones, represented in this part of the square, but it has troubles to distinguish the injured classes from each other. This may be the case because the diffusion properties are more related to the axonal loss in white matter present in both of these cases. A feature relevance analysis extract from the random forest reveals that fractional isotropy and axial diffusivity are the most important features to do this classification. This was kind of expected as Potter captures the general loss of structure in white matter. Orientation coherence and microfractional isotropy, metrics not accessible by diffusion tensor imaging, mildly contribute to the algorithm, but it do help to do the best classification. Both kurtosis sources, also not accessible by DTR, are the less relevant features to solve this problem. This might happen if these metrics have bias because the intrinsic assumption of the diffusion tensor distribution model that the microtensor are strictly Gaussian, that it's actually not the case for the generation. So in conclusion, this implementation suggests that it's possible to use machine learning algorithms and b-tensor encoding metrics to differentiate neurological tissue damage. While this work serves only as a proof of concept, the framework could generalize for other experimental settings, maybe in the future for clinical ones. Fractional isotropy and axial diffusivity are the more relevant features for this experimental setting. Orientation coherence and micro-FA improve the results from the algorithm, but not that much. Still, the best results are obtained using both pair of metrics. We emphasize that these results hold for these paradigms of white matter. Different settings like topological damage in gray matter or crossing fiber could have different weights. And actually, I'm just curious about these quantitative histology metrics that Amy showed you, on how to apply with these metrics and this data. So maybe I'll try that. Also, this is a published paper. You want to know more about the whole story? And thank you for listening. Happy to answer your questions.
Thank you so much, Ricardo. Any questions?
We don't have any question in the chat yet, but I have a question. So you said that FA and AD gives you the kind of the best metrics for differentiating between these pathologies. So do you and these two metrics can be used, can be extracted or estimated from like a normal diffusion protocol and not the b-tensor protocol. So do you think it's possible to get the same results with a normal diffusion protocol, diffusion encoding protocol, or you really need b-tensor?
No, yes, totally. At least for this experimental setting, in a perfectly aligned white matter track, you can only use diffusion tensor imaging to obtain these exact results. But that's why I was putting emphasis to that put here, because I would expect different, level priorities of the diffusion metrics for another experimental setting like crossing fibers or gray matter. I would totally expect that, picture to change in those settings. But for this case, yes, you can actually do this with using diffusion tensor imaging. The algorithms, it's a little worse, but not that much. So for practical purpose, maybe it's not necessary to go the full b-tensor encoding for perfectly aligned white matter degeneration, at least with this experiment.
Yes. Thank you. We have one question from Margarita Lopez. It is a very interesting work. How do you calculate the diffusion tensor density?
Sorry, the diffusion tensor what?
Diffusion tensor density.
Diffusion tensor distribution. With QTI plus with Q-space trajectory imaging, it's a method that you use to obtain metrics from a diffusion tensor distribution. You don't actually calculate the whole diffusion tensor distribution with this technique. You only obtain these metrics that gives you some approximation of how actually behaves the whole ensemble. There are actually works that try to infer the whole diffusion tensor distribution with this type of acquisition, but I'll say it's still an open problem that it's working out. So, yeah.
Okay. And in the continuation of this question, do you recommend some code for calculating diffusion tensor?
Yeah, I actually the things that I put in my slides, I put all the things that you actually need to do a proper b-tensor encoding acquisition in a Bruker scanner. You need to do the proper waveform optimization for a b-tensor encoding, but there are actually a lot of open resources in the web already, so you can use. I actually recommend to you start with the Q-space trajectory imaging. I would say the DTI equivalent for multidimensional encoding acquisitions with b-tensor, so it's a nice first step, and there are open code there for MATLAB, and it's actually in the IPy too, so there are already available resources on it.
Okay, thank you.
Thank you.
So I don't have any more questions.
Okay. All right. Big thank you again to all of our speakers.